Welcome to Dentospedia, your structured and simplified learning platform for various global dental board exams. And today I am to give you a short and high-yield lecture from the endodontic module, that is the embryology. Understanding embryology is essential because it tells us how the tooth forms, where each dental tissue originates, and why pulp, dentin, cementum, and PDL they behave the way they do clinically. In this chapter, we will learn each stage of the tooth development, the formation of the dental lamina, the key stage, the birth stage, the bell stage, and the cap stage. They give rise to the final tooth structure. So let's begin from the introduction. Now, before starting of the endodontics, we need to understand the pulp and the periradicular tissue, how they originate. So the pulp and the periradicular tissue, they originate from the ectomesenchymal cells. These ectomesenchymal cells, they help in the shaping of the tooth and they derive from the neural crest cells. These neural crest cells are the special cells, they originate from the nervous system, that is from the neural tube. And then they migrate into the face. And these cells, they give rise to dentin, pulp, cementum, and periodontal ligament. Now coming to the development of the dental lamina. This is the first step in the tooth development is the formation of the dental lamina. So dental lamina is the first step in tooth development. And it appears around the 6th week of the embryonic life. It is very important to know from exam point of view that it appears around the 6th week of the embryonic life. It is the thickened band of the oral epithelium. So it is the thickened band of the oral epithelium, and this band, they grow downwards into the underlying ectomesenchymal cells. From this, the dental lamina, that is the tooth bud, develops. And each tooth buds, they forms the both primary and permanent teeth. Coming to the embryology of the tooth development. Now the tooth development is also known as odontogenesis. The process of the tooth development is also known as odontogenesis. It is not a single event, but it occurs through the series or the well-organized or well-defined stages due to the interaction between the oral epithelium and underlying ectomesenchymal cells. So, the oral epithelium cells and underlying ectomesenchymal cells, they interact with each other to form the tooth development. And they are seen in the stages, that is the birth stage, cap stage, and the bell stage. So here in the diagram, we can see first, here is the oral epithelium and these is ectomesenchymal cells. They interact with each other to form birth stage, cap stage, bell stage, late bell stage, and this is how the tooth is formed. Coming first to birth stage, the first visible stage of tooth formation. Here the oral epithelium, they thickens. The oral epithelium, they thickens and they migrate into the underlying ectomesenchymal cells. And this epithelium outgrows, they enlarges to form the bud, marking the birth stage of the tooth development. So here in the birth stage, the tooth appears as a bud, hence it is known as birth stage. And these projection, each bud represents the future enamel organ. So each bud represents future enamel organ, very important, and this future enamel organ will eventually form enamel. And this marks the initial morphological stage of the developing tooth germ. So here in this birth stage, we understood it is the initial shape, it is the starting point of the enamel organ. The oral epithelium, they outgrows inward into the ectomesenchymal cells, and here the dental lamina is formed, and it is the future enamel organ. And here from the future enamel organ, enamel is formed. So this was it. Thank you so much. It was a short sample from the embryology chapter. To learn the complete chapter with diagrams, flowcharts, animation, and exam based MCQs, download the Dentospedia app today. Thank you.